Thank you very much. It is indeed going to be difficult for me to leave Stanford. This is a perfect place to be a scientist, I believe. I am going to talk to you, in order to try to get your attention, I'd like to talk to you about how to make babies. That might be something that you think you know quite a bit about, but I'd like to see if I can teach you a few things about it. I'll begin with the earliest days of development, and then I'll talk to you about how we're trying to change the outcomes of IVF. So on this slide, what you see is a series of pictures of human embryo development. Beginning in the upper left-hand corner, you can see a human egg. It's about the size of a quarter of the point of a pin. And on day 1, this egg has been fertilized, and you can see that there's 2 nuclei, a maternal pronucleus carrying the mother's chromosomes, and a paternal pronucleus carrying the father's chromosomes. These two pronuclei migrate towards each other, and as they're migrating, the DNA begins to be reprogrammed. It's erased of all kinds of marks that are on the chromosomes, and the embryo begins to reprogram from being egg and sperm to being an embryonic cell fate. Following day 1, you can see that the embryo begins to go through a series of cleavage divisions. It's gone from 1 cell to 2 cells. Later on day 2, it goes from 2 cells to 4 cells, and then from 4 cells to 8 cells. And it's still the same size, about a quarter of a size of a point of a pin. Now on day 3, for the very first time, the embryo turns on its own genes. Before that, it was primarily running off of programs it inherited from the egg and the sperm, but for the first time, the reprogramming is done, and the embryo turns on those unique human embryonic genes that really define us. On day 4, you can see that the embryo goes from 8 cells. It now has about 16 to 32 cells, but the embryo has collapsed, it's compacted. And with the act of compaction, what happens on day 5 is the cells on the outside form trophectoderm. They're going to attach the embryo to the uterus if it's in a woman. And the cells on the inside, shown at this light arrow here, go on to form the child, okay? Now, when you think about this process, I'm sometimes asked, how did you get interested in this? But to me, it's just amazing. You go from a sperm and an egg to 1 cell, 2 cell, 4 cell embryo, and there's a program here. Now, following day 5, the embryo, if it's in a woman, will hatch. It'll come out of its shell, attach to the uterus, and begin to grow. Human embryonic stem cells are derived from these inner cell mast cells at the light arrow. And what that means is that since these cells can give rise to the entire child, human embryonic stem cells have the potential to give rise to all the different cell types of our body. Excuse me. So what I want to stress is that most of what we know of the first 6 days of human embryo development, we've learned from embryos that are cultured in a dish for the purposes of in vitro fertilization. And in the United States, in general, there's about 1.5 to 2 million embryos produced each year, and nearly half of those are discarded. So about 800,000 or so are discarded. Out of that 800,000, it's probably fewer than 1,000, maybe even fewer than 500 that are used for research each year. So it's a minuscule number of embryos that are used. Now, as you know, there are regulations, federal regulations against embryo research. And these have had some real impacts on our understanding of human embryo development. Our understanding is very, very poor, and this has some consequences. So the number 1 cause of birth defects is errors in the human embryo, primarily chromosomal abnormalities, imprinting disorders, translocations. Reproductive failure is very, very common, so that 10 to 15% of couples are infertile. Embryo loss and miscarriage is very, very common in the human population. And associated with in vitro fertilization, there are two really troubling things. One is that nearly 40% of births are multiple births, meaning that there's twins or triplets or greater, and the multiples often have great complications. From prematurity, low birth weights, cerebral palsy, there's a number of complications to multiples. And then in some cases, because so many embryos are transferred, because it's difficult to tell which embryo is viable, there's a need during the pregnancy for fetal reduction. And that's exactly as it sounds. There's a need to remove one or more of the fetuses as they're developing because the woman simply cannot carry the fetuses. So when I first came to Stanford, we decided we were going to make an atlas of human embryo development. And this atlas or map would include imaging data, molecular pathway data, what genes are on, what genes are off, genetic and epigenetic data. And so what we did is shown on this slide, beginning on number 1. We thawed some one-cell embryos. One of my first graduate students was a mechanical engineering graduate student, and I'm not exactly sure why he joined my lab, but he decided to make these little microscopes that are dark field, they're called, and they fit into incubators that grow embryos. Human embryo development, not all the embryos reached that blastocyst stage and hatched. And so the trick in IVF is determining which embryos will go on to develop and which are going to arrest. They have to be transferred early. And so in the year 2010, we published a paper that included this figure entitled The Fundamentals of Human Embryo Development. The year is 2010, and we're describing the fundamentals. And what we found was that we could determine which embryo would make it with greater than 93% sensitivity and specificity by watching the movies and watching how long an embryo took in the first cytokinesis. So if this is one cell, you see a little furrow begin, and it should be 15 minutes plus or minus 5 minutes from that time of formation of the furrow to 2 cells. From 2 cells to 3 cells should be 11 plus or minus 4 hours, and the third and the fourth cell should appear almost synchronously. Now, this data was in great contrast to what was expected. In the clinic, people thought that embryo development was rather variable. If you watch the movie, it seems like different embryos are doing different things. But successful human embryo development is very precise. And this allowed us to then write computer programs. I'll show you the second one. Write computer programs that could model human embryo development and predict which embryo was actually going to succeed. That we can model human embryo development and determine which embryo is actually most likely to develop. And I won't, because of the hour is getting rather late, I won't go into a lot of the details, but we learned a lot about what these behaviors mean. And they're very closely related to whether or not embryos have the right chromosome number when they begin development. So almost 70% of human embryos have the wrong number of chromosomes. The most common chromosomal abnormality is, of course, Down syndrome in the human population, but most miscarriages are caused by an abnormal number of chromosomes. And so I won't go into all of my models and all of my slides, but just let me show you one other slide, and that is in the year 2012, we were able to come up with a new model for how human chromosomal abnormalities occur early in embryo development. And in 2013, we had 2 results that were really, really gratifying to me. The first on the left-hand corner shows an article from the Herald of Scotland, a company called Oxygen, of which I'm a founder, licensed our technology and developed an assay called the Early Embryo Viability Assay, or EVA. The first child that was conceived was a little girl, and she was named Eva, E-V-A. The first child born was born premature on the right-hand side, but if you actually look at this article, it is very touching. The parents had been trying for 8 years to have a child. The mother had been miscarrying. She had had 6 or 8 miscarriages, and they used our technology to pick out an embryo that might be viable. And so with that, I'd really like to summarize more broadly, stepping back, that it would be a shame not to really move forward in science at this time in history with all we have. It's a very unique time in scientific history with many tools and many resources at our disposal for use that we previously did not have. We are hopeful that our results will provide a foundation to understand human embryo development. We think it's important to give the next generation big scientific problems to consider. And one of the biggest problems, if you ask me, to consider is how does an egg and a sperm come together and we ultimately get a child and an adult, and what is associated with healthy versus unhealthy or poor outcomes. And our results are being currently translated to the clinic. And so with that, I would acknowledge people that have collaborated with me at Stanford. I apologize for my voice. Something about this is making me a little bit misty. I don't know. Stanford has been a very, very, very special place to me. And I'd love to stress the funding from CIRM that we have received from the California Institute for Regenerative Medicine. Our studies are not fundable under any circumstances by federal funds. And yet, if you think about them, if you think in a rational world, you can reduce the need for fetal reduction, multiple births, poor outcomes, and especially help families. I acknowledge the March of Dimes, the Stanford School of Medicine. Phil is gone, but he was instrumental in supporting us, an anonymous donor. And then I do thank Oxygen, because the private companies can bring your technology to the clinic in a more efficient way in some cases like ours. So thank you very much.